This lecture is on Chapter 2, the chemical composition of the body. Chapter 1, Section 1 starts with the atoms, ions, and chemical bonds, and they actually start with the same thing I tell my students in class, that since physiological processes are based on chemical reactions, a general understanding of chemical principles is necessary. Students always wonder, why do we have to look at chemistry in a biology class? But in every single biology class I've ever taken or taught, chemistry has been an essential part, because chemistry makes the foundational unit of life's structure. So we'll start with atoms. An atom is the smallest unit of an element. It has a nucleus with positively charged protons and uncharged neutrons. It has orbiting electrons with negative charges. An atomic mass equal to the number of protons plus the number of neutrons gives the atom mass, and the protons and the neutrons contribute to it, but the electrons have negligible mass. Nonetheless, electrons are very important, and we'll talk about them more. The atomic number is equal to the number of protons. You're going to learn that the number of neutrons might change in the nucleus, and that results in isotopes. And you'll learn that the number of electrons might change in the orbits around the nucleus, and that makes an ion. But the number of protons must stay constant, or else the atom is now something completely different. This structure shows a hydrogen atom and a carbon, and that is your figure 2.1 in your book. It says that on the left, the electron shells are represented by shaded spheres indicating probable positions of electrons. On the right, the shells are represented by concentric circles. So if you notice that the hydrogen has one proton and one electron, if that hydrogen gained a proton, as can happen in nuclear fission, like in the sunshine, it is no longer a hydrogen. It's something completely different. It is now a helium. So that illustrates that the protons are important in determining what the atom is. So the number of protons indicates the atomic number and what that atom is. So hydrogen will always have one proton. The electron can change, however, and it can actually kick that one electron off and become just a positively charged proton, basically. Or it can form covalent bonds and share another electron with another atom. The carbon is 6 protons, 6 neutrons, 6 electrons. And so the same rule applies as the atomic number has to be the protons. Carbon has 6 neutrons in its natural state, but it can actually gain neutrons and become an isotope. It can actually become a heavier version of itself. I like to think of that being kind of the normal aging process of a person. You can become heavier as you get older. Anyway, and then the electrons, you see that there's 4 electrons in that outer shell of carbon. So in order to balance its number, it generally will form covalent bonds. So the electrons are very important in bonding behavior. Protons are very important in determining what atom we're looking at, and the neutrons are important in determining whether we have an isotope. So let's look at these electron orbitals. Electron orbitals, or shells, are energy levels that surround the nucleus of an atom. Electrons fill the shells, starting with the one closest to the nucleus. The first shell generally holds 2 electrons, and each shell thereafter generally holds 8 electrons. Non-biological elements fill distant shells that hold more than 8. If you hear any scraping noises, my apologies. It's my kitty cat. All right. Atoms are most stable when the outer shell is filled. Electrons in unfilled outer shells participate in bonding, and they are called the valence electrons. Table 2.1 features atoms that are commonly present in organic molecules, and it's illustrated in blue in your book and that peach color here. And so we see hydrogen, carbon, nitrogen, oxygen, and sulfur. In a lot of my classes I teach, a common exam question is, what are the 4 major atoms found in the body? And your answer to that would be the hydrogen, carbon, nitrogen, and oxygen. Isotopes are atoms of the same element but with different numbers of neutrons, like I had said with the carbon earlier. Their atomic number is the same, but the atomic mass is different. My apologies. So isotopes, as I mentioned, are atoms of the same element but with different numbers of neutrons. Their atomic number is the same, but the atomic mass is different. Some isotopes are radioactive and used in medical testing and physiological research. The term chemical element includes all of the isotopes of that element. Okay, so back to these valence shell electrons. We're going to talk about chemical bonds. Chemical bonds are formed when electrons in atoms interact. The number of bonds an atom can form is determined by the number of valence electrons. Hydrogen is 1 electron, and it needs 1 more to fill the inner shell so that it can form 1 bond. Carbon has 6 electrons, 2 fill the inner shell, and 4 are valence electrons. It needs 4 more electrons so that it can form 4 bonds. We also learned that sometimes hydrogen can kick an electron off and basically exist as a free proton. This makes a hydrogen very important in acids and bases. We'll talk about those in a little while. Okay, so let's go in and look at the different types of bonds. The first one is the covalent bond. Valence electrons are shared because energy levels overlap. So in a covalent bond that is nonpolar, the electrons are shared equally. For example, 2 hydrogen bonds. In polar covalent bonds, electrons are not shared equally, and thus they have positive and negative ends. An example is the bond between oxygen and hydrogen in water. So this figure, excuse me, kitty cat. This figure 2.2 shows a hydrogen molecule with covalent bonds between the hydrogen atoms. And this is a nonpolar covalent bond. In figure 2.3, we see the molecules methane and ammonia represented in 3 different ways. It says, notice that the bond between 2 atoms consists of a pair of shared electrons, and the electrons from the outer shell of each atom are involved. These are also looking like stable non-polar bonds. When we get to water, that's that polar bond discussed in the last couple of slides. And I'll explain a little bit more how that works. I wanted to get to a picture for you first. All right, so water, of course, is that polar molecule, meaning that it's going to have like a slightly charged end that'll be positive and a slightly charged end that'll be negative. And because of this polarity, it makes water a good solvent. Substances will dissolve in it. Water also can form a strong hydrogen bond. And the polar molecules that are made up of covalent bonds may dissolve in water and are called hydrophilic. Ow! Good Lord. Struggling tonight. Hold on just a second. Okay. So we're talking about how water can form strong hydrogen bonds, and polar molecules may dissolve in water and are called hydrophilic. So that word you need to know for your study guide for sure. So I wanted to make sure to cover that. So hydrophilic, if you look at that word, means fond of water, water loving. And then nonpolar molecules do not dissolve in water, and they are called hydrophobic. So phobic means fearing of water, hydrophobic. Figure 2.4 shows a model of the water molecule showing its polar nature. So you can see the positively charged ends here on the hydrogen and the slightly negative charge for the oxygen. So it goes on to say, notice that the oxygen side of the molecule is relatively more negative, whereas the hydrogen side is relatively more positive. Polar covalent bonds are weaker than nonpolar covalent bonds. As a result, some water molecules ionize to form a hydroxide ion and a hydronium ion, a hydrogen ion, shown here on the right side. We see the hydroxide, which is OH negative, and the hydrogen, which is H positive. So the oxygen is what we call electronegative, and your book states that atoms of oxygen, nitrogen, and phosphorus have a particularly strong tendency to pull electrons toward themselves and are thus electronegative when they bond with other atoms. And so they tend to form polar molecules. Okay, ionic bonds are our next type of bond. In an ionic bond, one atom gives electrons to another so that both have filled valence shells. The electron donor becomes positively charged, and the ion is called a cation. The electron receiver becomes negatively charged, and the ion is called an anion. Cations and anions attract each other to form ionic compounds. And as far as I understand, once an ion is formed, it's generally going to stay in its ionic state. And so when you think of a typical sodium atom, you don't usually see it as this top form that's featured in figure 2.5. In this top form, you can see that the sodium has one electron in its outer shell and thus is highly reactive. So if you had pure sodium, that would be a very highly reactive atom, and it tends to actually combust when it interacts with water. So once the sodium has rid itself of that extra electron and stabilized, then as far as I understand, it stays in this ionic state. And the sodium ions and the chloride ions make up a large portion of what we call electrolytes in our body, and that helps to balance our intracellular and extracellular fluids, and it also serves to power electrical impulses and nerve impulses and muscle contractions. So most ionic compounds will dissociate or come apart with no reaction when dissolved in water. So it's the charges that are holding these atoms together, because as I mentioned earlier, once the sodium atom has gotten rid of that extra electron, it could have done that billions of years ago, you know? And so that positive charge of the sodium ion and the negative charge of the chloride ion is what's going to be attracted to each other and hold that bond, kind of like magnets. And that's why when ionic compounds are put in water, they dissolve because of that polarity of the water molecule. So the negative side of water is attracted to the cation, and the positive side of water is attracted to the anion. So in the case of the sodium and the chloride, we'll see that in the next illustration, figure 2.6, and you'll see these spheres of hydration surrounding the sodium and the chloride ions, pulling those ions apart. And so again, these free ions are critical to many physiological processes, and they're termed electrolytes. Here's figure 2.6, how sodium chloride dissolves in water. So again, the negatively attracted oxygen is, the negatively charged oxygen is going to be attracted to the sodium cation, and the positively charged hydrogens are going to be attracted to the chloride anion. And other water molecules are attracted to this first concentric layer of water, forming hydration spheres around the sodium and chloride ions. The polarity of water allows for water molecules to bond to each other through those hydrogen bonds mentioned earlier. So let's look at the hydrogen bond in more detail. Hydrogen bond is the third type of bond we've talked about. We talked about covalent, which include the polar and nonpolar. We talked about ionic, and now the third is the hydrogen bond. So the hydrogen bond is a weak bond formed between two polar molecules based on opposite charges attracting. It's not based on electron sharing. Hydrogen bond forms between electropositive hydrogen atoms and an electronegative oxygen or nitrogen. It forms between water molecules and is responsible for surface tension and capillarity. Hydrogen bonds forms between amino acids on a protein to produce the 3D structure of the protein. And the hydrogen bonds hold the two strands of the DNA molecule together. Now, these bonds are easily broken, but they're also easily and readily made. That ability to break and form very easily is shown by this dashed line. You can see the water molecules bonded to each other through these hydrogen bonds. The electronegative oxygen, electronegative means slightly negative, it's got that pull to the electrons. It's going to be attracted to the electropositive hydrogen of the other water molecule. So when the water molecules are bonded together, that's called cohesion, and it gives water the property of surface tension. And it also can lead to other properties such as adhesion, which is water molecules sticking to other substances, such as the sodium and the chloride, and then once they pull those apart in the spheres of hydration, they form those electrolytes. Okay, moving on to the acids, bases, and the pH scale. So this is where those hydrogen ions are going to come into play. Some water molecules will ionize to form free hydrogen ions or protons, the hydrogen plus, and the hydroxide ion, which is an OH negative. When this happens, there are the same number of hydrogen ions as hydroxide ions in solution. And this is a neutral solution, which is said to have a pH of 7, and that means 10 to the negative 7 molar concentration of hydrogen ion. This is a inverse logarithmic scale, the pH scale, and we'll talk about it being an inverse scale in a moment. So when a solution has more hydrogen ions than hydroxide ions, this is called an acid, and the pH is below 7. Acids are often called proton donors. So this is that inverse relationship that I just mentioned. When we have more hydrogen ions than hydroxide ions, the pH is low. So the higher the hydrogen ion concentration, the lower the pH. So acids have a very low pH and a lot of hydrogen ions. When a solution has more hydroxide ions than hydrogen ions, this is called a base, and its pH is above 7. Such solutions are also called alkaline, and a base is often called a proton acceptor. So again, an inverse relationship here. When we have less hydrogen ions and more hydroxide ions, the pH is higher. So a high pH indicates less hydrogen ions and more hydroxide. So this is the pH scale, which, as mentioned, is a logarithmic equation. And so the pH scale runs from 0 to 14, with 0 being the strongest acid and 14 being the strongest base. And so there's the equation. It's a log of 1 divided by the molar hydrogen ion concentration. And it can also be expressed, it says, as pH equals the negative log of the hydrogen ion concentration. Pure water has a hydrogen ion concentration of 10 to the negative 7, so the pH is 7. And in your book it says, because of the logarithmic relationship, a solution with 10 times the hydrogen ion concentration, which is 10 to the negative 6 molarity, has a pH of 6. So a pH of 6 has 10 times the number of hydrogen ions as the pH of 7. Whereas a solution with 1/10 of the hydrogen ion concentration, 10 to the negative 8 molarity, has a pH of 8. So it has 10 times less hydrogen ions than a pH of 7. It says the pH value is easier to write than the molar hydrogen ion concentration, but it is admittedly confusing because it is inversely related to the hydrogen ion concentration. Buffers are our next topic. So buffers stabilize a pH in a solution. Buffers have two components, a weak acid and a weak base, and that is a buffer pair. In blood, two molecules stabilize pH, the bicarbonate ion, HCO3 negative, and carbonic acid, H2CO3. In step 1, we see the equation, and it's the bicarbonate ion plus a hydrogen ion yields carbonic acid. And it says the double arrows indicate that the reaction could go either to the right or the left. The net direction depends on the concentration of molecules and ions on each side. If an acid, such as lactic acid, should release hydrogen ions into the solution, for example, the increased concentration of hydrogen ions would drive the equilibrium to the right, and the following reaction would be promoted. We would go towards making more carbonic acid because the bicarbonate would accept or soak up the excess hydrogen ions. Thus, bicarbonate neutralizes excess acids, and then carbonic acid in turn will neutralize excess bases. If the blood falls below a pH of 7.35, the condition is called acidosis. If blood rises above pH of 7.45, the condition is called alkalosis. The action of the bicarbonate carbonic acid buffer system, as well as the lung and kidneys, work together to prevent acidosis or alkalosis. Organic molecules contain carbon and hydrogen. Because carbon must form 4 bonds to satisfy the valence shell, it can form chains and rings of carbons while still bonding with other atoms. This is why carbon is considered the backbone to life, and we're carbon-based organisms because of its ability to form those chains and rings while still bonding with other atoms. 2 carbons can share 1 or 2 pairs of electrons. If 2 pairs are shared, it is a double bond and can bond with 2 additional atoms. A double bond is also stronger than a single covalent bond. If 1 pair of electrons are shared, it can then bond with 3 additional atoms. Illustration 2.8 shows single and double covalent bonds, and 2 carbon atoms may be joined by a single covalent bond, shown on the left, or a double covalent bond, shown here on the right. In both cases, each carbon atom shares 4 pairs of electrons, thus has 4 bonds, to complete the 8 electrons required to fill its outer shell. Hydrocarbon structures. Carbons are not shown in the molecular structures. Like here, we see the ring, and where you see the corners here, that indicates a carbon. So carbons are not shown but are understood to be at the corners of a molecule. And some show double bonds, as featured here in the benzene, that shows the double bond between these 2 carbons. Carbon chains and rings form the backbones for more reactive groups of atoms called functional groups. Functional groups allow organic compounds to be classified into groups, which provides unique chemical properties. Okay, so we just seen this picture showing the corners indicating where the carbon would be, and now you know where the double bonds would be, and then the top picture A is going to be linear. It's like a chain. B, it says, is cyclic, and then C is an aromatic ring. Figure 2.10 shows the various functional groups of organic molecules. The general symbol for any functional group is R. Specific functional groups are indicated within the gold rectangles. So we see carbonyl, hydroxyl, sulfhydryl, amino, carboxyl, phosphate. And then we see categories of organic molecules based on functional groups on the right here, which is figure 2.11. We've got acids, alcohols, and other types of organic molecules characterized by specific functional groups. Stereoisomers are molecules that have exactly the same atoms arranged in exactly the same sequence, but they differ in the spatial organization of their functional groups. This characteristic is critical to function. A given enzyme may interact with one stereoisomer but not another. The sugars glucose, galactose, and fructose are stereoisomers. You may have heard of isomers before, but this book goes into more detail with the term stereoisomers. And so this illustration is figure 2.13, and we see that in A up here, stereoisomers include molecules such as butene, which can have its methyl group CH3 on either the same side, which is cis, or opposite sides, which is trans. And in B, we see that other stereoisomers are enantiomers, which are optical isomers, which are mirror images of each other. Using the molecule glyceraldehyde as a reference, these can be designated as either D or L isomers. And so it's kind of fun to see where the mirror image comes into play, and it's this hydroxyl group that you see between D and L. And you've heard of this before because you might have heard of like L-arginine or D-mannose. And so now you know that it's talking about those enantiomers, which are the mirror images of the isomers. Now that we've looked at the way carbon can bond and the functional groups that can attach to carbon structures, let's start taking a look at the four major biological molecules. They're going to be our carbohydrates, lipids, proteins, and nucleic acids. So the book groups the carbohydrates and the lipids in the same section. So we'll talk about those first. For carbohydrates, the characteristics include the fact that they are organic molecules that contain carbon, hydrogen, and oxygen in a 1-to-1 ratio. So you may have heard C6H12O6 for glucose. So 6 carbons, 6 times 2 would be 12 hydrogens, and then 6 oxygens. So carbohydrates serve as a major source of energy in the body, and they include sugars and starches. The suffix OSE denotes sugars. So earlier when I said D-mannose, now you say, oh, that's a sugar. Yes, lactose, glucose, sucrose, fructose, galactose, all of those, os. Another term that denotes a sugar is saccharide. So in the classifications of carbohydrates, we have monosaccharides, disaccharides, and polysaccharides. Saccharin is the Greek root word for sugar. So monosaccharide is a simple sugar, a one-carbon ring. Examples are glucose, fructose, and galactose. And that structural formula of C6H12O6 leads to isomers, as mentioned earlier. The disaccharide, di for two, is two monosaccharides joined by a covalent bond. Examples are sucrose, maltose, and lactose. This is figure 2.14, and it shows the structural formulas for 3-hexose sugars. So these are, hexa means the six carbons, hexa. And then we have like pentose sugars, which will have 5 carbons when we look at like nucleic acids. So these are hexose sugars, 6 carbons. So let me read this. We have A, glucose, up at the top, B, galactose, and C, fructose. All 3 have the same ratio of atoms, C6H12O6. The representations on the left more clearly show the atoms in each molecule, while the ring structures on the right more accurately reflect the way these atoms are arranged. If you ever take a organic chemistry class, you'll get to build molecules like this. Okay, polysaccharides. Polysaccharides are going to have several monosaccharides, generally glucose, joined together. Common polysaccharides include starch, glycogen, cellulose, and chitin. Starch is the plant storage of sugar, composed of thousands of glucose molecules. Glycogen is the sugar stored in animal cells. It's similar to starch, but more highly branched. Cellulose makes up the cell walls of plants. Cellulose is not digestible by humans. And chitin forms the exoskeleton of arthropods. It's similar to cellulose, but it has amine groups in the glucose subunits. Generally, when you eat cellulose, it's called fiber, and we have soluble and insoluble fibers. Both are beneficial in the diet. The soluble, I believe, is what lowers cholesterol, and the insoluble, I believe, is what forms bulky stool to help you pass your stool. Starch, you know of with rice and bread. That is how the plant stores its nutrients that it needs, and humans have figured out how to cultivate that so we can use it for our food. And then glycogen is how animals store their sugar in like the liver and the muscles. You get a good appreciation for that branch structure of glycogen in this image, figure 2.15. So it says glycogen is a polysaccharide composed of glucose subunits joined together to form a large, highly branched molecule. When it elaborates a little bit more in the book about the starch and the glycogen, says that animals have the enzymes to digest the bonds between adjacent glucose subunits of starch and glycogen, but we do not have the enzymes to digest the cellulose, hence the fiber. And this is important. As far as cells storing the carbohydrates for use as an energy source, if many thousands of separate monosaccharide molecules were stored in a cell, their high concentration would draw an excessive amount of water into the cell, damaging or even killing it. The net movement of water through membranes is called osmosis, and cells that store carbohydrates for energy minimize this osmotic damage by instead joining the glucose molecules together to form the polysaccharide starch or glycogen. Because there are fewer of these larger molecules, less water is drawn into the cell by osmosis. So this gives the body of both the plants and animals a challenge. The plant has to break down its starch itself to use it for energy, and the animal has to break down glycogen to use it for energy. And so this is done through dehydration, synthesis, and hydrolysis. So covalent bonds that hold monosaccharides together are formed via dehydration synthesis, where a hydrogen atom is removed from one molecule and a hydroxyl group is removed from another to form water. Hydrolysis breaks bonds between monosaccharides, it adds water and splits the molecule. And both of these, dehydration synthesis and hydrolysis, use enzymes to accomplish this feat in the bodies of animals such as ourselves, and then, of course, plants for the starches. So dehydration synthesis and hydrolysis can also employ different enzymes to build and break down fats, proteins, and nucleic acids. This is illustration 2.16 in your book, and it shows the dehydration synthesis of disaccharides. So the two disaccharides formed here are A, maltose, and B, sucrose, also known as table sugar. Notice that a molecule of water is produced as the disaccharide is formed, and that's why we call it dehydration synthesis, because when you take the hydrogen off of one monosaccharide and the hydroxyl group off of the other monosaccharide to form this disaccharide, you end up joining them with the leftover oxygen, and the water then is released. The opposite of that is hydrolysis. So you add water to the system, and that's what hydrolysis breaks down to, meaning, no pun intended, breaks down. Haha. Okay, anyway, so hydro is the water, lysis means to cut or snip or break. So you add water to break the molecule apart. So this is the hydrolysis of starch. The polysaccharide is first hydrolyzed into disaccharides in A up here. And the disaccharides are maltose. And then they are further hydrolyzed to the monosaccharides of glucose. So we've got 2 glucose as our end products. Notice that as the covalent bond between the subunit breaks, a molecule of water is split. In this way, the hydrogen atom and the hydroxyl group from the water are added to the ends of the released subunit. So basically, we've just gone back to where we started with the hydroxyl groups in those corners of the glucose. If we go back here to figure 2.16, where the glucose molecules are joined, you can see that hydroxyl groups join to each other. I always think of OHO. Sorry about that. OHO. And so then we end with OHO once more when we do hydrolysis and split those glucoses back. So again, it's important to build the storage molecules so that we don't kill ourselves with osmotic pressure. But then we have to break those sugar molecules back off in order to then use them in our cells. And so that will be where hydrolysis comes into play. And this doesn't just magically happen. An enzyme is involved in every step. Let's move on to lipids now. So lipids consist of non-polar hydrocarbon chains and rings that makes them hydrophobic. Thus, lipids are insoluble in water. Carbohydrates, which we just talked about with that C6H12O6, those are soluble in water. So carbohydrates are like polar and hydrophilic. Lipids are non-polar and hydrophobic. There are several categories of lipids. The first category is the triglycerides or triacylglycerols. That's a new word for me. So the triglyceride is a subcategory of lipid that includes the fats and oils. So fats are the solids and oils are the liquid forms. So like saturated versus unsaturated fatty acids. And that's what the triglycerides are composed of. They're composed of one molecule of glycerol and three molecules of fatty acids. Glycerol is a 3-carbon alcohol. And it says because of this structure is why chemists currently prefer the name triacylglycerol, although the name triglyceride is still in wide use. That's what we're going to use because that's what it is on our medical reports. Blood triglycerides. Okay. So the fatty acid structure. The fatty acid is a long, non-polar hydrocarbon chain with a carbonyl at one end, hence the fatty acid. The carbonyl is the acid part. I always think "coo" when I see an acid, COOH, coo. If every carbon on the fatty acid chain shares a single pair of electrons, that fatty acid is saturated. If there are double bonds between carbons, the fatty acid is unsaturated. These are called neutral fats when they're stored in adipose tissue. Trans fats are artificially produced straight-chained fatty acids made from vegetable oils. Like partially hydrogenated oils are like trans fats. And there's a big write-up in your book on the lifestyle application of trans fats. They behave like saturated fats in the body and can lead to heart disease. Figure 2.18 shows the structural formulas for fatty acids. In A, the formula for saturated fatty acids with the single bonds, and so this would be straight across and solid at room temperature. And then B is showing where the double bonds are, and those are the points of unsaturation, and they're highlighted in yellow. And so we call these the unsaturated fatty acids. And sometimes they like curl. So it's showing it linear here in this illustration. But in reality, boom, we see that it can curl and bend and all that. And so here is the cis to trans fats in figure 2.19. Says oleic acid is a naturally occurring fatty acid with one double bond. Notice that both hydrogen atoms in yellow are on the same side of the molecule, and this is called the cis configuration. The cis configuration makes this naturally occurring fatty acid bend. The fatty acid on the right, however, is the same size and also has one double bond, but its hydrogens here are on the opposite side of the molecule, and this is called the trans configuration. This makes the fatty acid stay straight, more like a saturated fatty acid. Note that only these hydrogens and the ones on the carboxyl group are shown. Those carbons that are joined by single bonds are also each bonded to two hydrogen atoms, but those hydrogen atoms are not illustrated. Figure 2.20 shows the formation of a triglyceride molecule from glycerol and 3 fatty acids by dehydration synthesis reactions. And you can see the OHO that I was mentioning earlier. And so since this is dehydration synthesis, you can guess that each one of these OHOs is going to yield a water molecule, and that's what we get at the end here, the triglyceride bonded together with its fatty acids and glycerol head and the 3 waters. And then what's left over after we've removed those waters is that oxygen that will then be bonded to the carbons. Ketone bodies. Ketosis is a hot topic these days, and ketosis is going to occur from the formation of ketone bodies. Ketone bodies are formed by the hydrolysis of triglycerides, and this forms the free fatty acids in the blood, And those fatty acids can be used for energy or converted into ketone bodies by the liver. Strict low carbohydrate diets and uncontrolled diabetes can result in elevated ketone levels called ketosis. Ketone levels that are high enough to lower pH can cause ketoacidosis, which can lead to coma and death. Like diabetic coma is usually ketoacidosis related. So a sign of ketoacidosis is acetone breath. When I think of acetone, I think of nail polish remover. Figure 2.21 shows ketone bodies. Acetoacetic acid, an acidic ketone body, can spontaneously decarboxylate, which means lose carbon dioxide, to form acetone. Acetone is a volatile ketone body that escapes in the exhaled breath, thereby lending a fruity smell, more the odor of nail polish remover, to the breath of people with ketosis, which is elevated blood ketone bodies. So the keto diet is not like a forever diet. It's a very brief diet that is for rapid weight loss, and then you should incorporate carbohydrates slowly and limitedly back into your diet. Phospholipids are the next group. This will be the third group. We had triglycerides and then ketone bodies. So now we have phospholipids. I love these ones because these are the ones that are going to make up the cell membranes. So phospholipids are lipids with a phosphate group, and it makes them part polar and part nonpolar, which is called amphipathic. And amphi means like between states. So amphibians like frogs and salamanders live part of their life in water, part of their life on land. And so amphipathic molecules are partly polar and partly nonpolar. And this is important because as that major component of cell membranes, it's going to end up forming a double layer. The hydrophilic phosphates are going to point outward on each side, and the hydrophobic fatty acids and glycerol will point inward. And this makes a double layer. As micelles, phospholipids can act as surfactants, and the polar nature of the molecule decreases the surface tension of the water. And so this surfactant keeps lungs from collapsing. So phospholipids acting as surfactants is very important in the lungs, and also micelles can transport fats through the bloodstream. Okay, so we are looking at figure 2.22, and this is the structure of lecithin. Lecithin, also called phosphatidylcholine, is where choline is the nitrogen-containing portion of the molecule. Interesting, choline is also part of an important neurotransmitter known as acetylcholine, discussed in Chapter 7. The detailed structure of the phospholipid top is usually shown in simplified form on the bottom. Yep, this is how I was drawn on the board, the bottom one with the phosphate head and the fatty acid tails. And the circle represents the polar portion, which is PO4 negative. And then the sawtooth lines, is a better word than mine, squiggly. The sawtooth lines show the nonpolar portion of the molecule. So those would be the hydrocarbons. And, the phospholipid generally has a fatty acid that is saturated and a fatty acid that's unsaturated, which causes them to be slightly oily. Figure 2.23 shows the formation of a micelle structure by the phospholipids such as lecithin. The hydrophilic outer layer of the micelle faces the aqueous environment, so you can see where the water would be, and this inside, as it had mentioned, the micelles can help you to digest and transport fats in the blood and then make them a surfactant. That's what I wanted to remind you of. And it helps to prevent the lungs from collapsing. Moving on to the fourth type of lipid is the steroids. There are a lot of different kinds of lipids. We have one more in the book after this one. So number 4 is the steroids. These are waxy-like in their structure. They've got these really neat looking rings. And so cholesterol is going to be a steroid lipid, and we generally think of cholesterol as a bad thing, but it's not. It's a precursor to the steroid hormones such as testosterone, estrogen, and the corticosteroids, and it also is used to make molecules such as vitamin D and bile salts. Another thing cholesterol does is it helps stabilize the cell membrane, because remember I told you that the phospholipid tends to have like a saturated fatty acid and an unsaturated fatty acid, making it slightly, liquid. And so the cholesterol in there will help stabilize the cell membrane, making it a little more waxy, a little more solid than it would be otherwise. Let's look at the other points on this slide. The steroid is structurally very different from a triglyceride, but is nonpolar, so it is considered a lipid. And so those 3 carbon rings plus 1 5-carbon ring plus a functional group makes that steroid. And when we see that cholesterol, that little tail that's up here where it says like the C27 cholesterol, that little tail can interact kind of think one of those ends is what sits in the cell membrane. It might be the hydroxyl end. So let me not get myself in trouble and get ahead of myself here. We'll wait and look at which end is stuck in the cell membrane when we get there. Let's see if it actually shows it. Just real quick, out of my own curiosity, my apologies. Be going too deep, but it seems to me that whenever I've taught about it before, it's that tail at the end that's in the fatty acid portion of the phospholipids, and then this kind of ring portion with the OH tends to be near the phosphate, but I'm not seeing it, and so I'm going to move on so as not to waste your time. I apologize. Let me get back to my original page and we'll go forward. Okay. I just love the fact that it sits in the cell membrane and helps to strengthen it. It's just a neat feature of cholesterol for me, because before teaching the anatomy classes and the physiology classes, etc., cholesterol always just kind of had a bad rap. But of course we know that there's the LDL cholesterol, the HDL cholesterol. HDL is the healthier version, usually transporting fat out of the body. LDL is the one that is the low density lipoprotein, so it's got the one that's got more fatty acids and can lead to the heart disease, etc. And then your overall blood cholesterol matters as well. I'm watching mine because mine's a little high. I ate butter for a long time, and it raised my cholesterol. And not only butter, but I ate a lot of cheese too. And let's just admit it, I was finally making money, so I was eating all steak. So anyway, whoops. Okay. But anyway, the last one. Prostaglandins are a type of fatty acid with a cyclic hydrocarbon group, and this is really interesting because they serve as regulatory molecules between cells in the same organ. So they almost act like local hormones that don't travel through the bloodstream. And they help regulate blood vessel diameter, ovulation, uterine contractions, inflammatory reactions, blood clotting, and many other functions. So you may have heard of prostaglandins before, particularly with the inflammatory reactions. They can raise that. Figure 2.25 shows the structural formulas for various prostaglandins, and they are a family of regulatory compounds derived from a membrane lipid known as arachidonic acid. I always like saying that word, arachidonic. Okay, proteins. Proteins are going to be made up of an amino acid chain. It's that R group that is going to make them so diverse. So an amino acid has an amino group, a carboxyl group. So the amino is like the nitrogenous group. The carboxyl group is going to be the acidic group, amino acid. And then the functional R group is what gives the amino acids their particular features. And so there are 20 different amino acids that can be combined in an endless number of ways. Just like our alphabet has 26 letters and we can do an endless amount of words with that, 20 amino acids can make an endless amount of words. And so the functional group, as I mentioned, is what differentiates those 20 amino acids. It's indicated by the R. And amino acids can be polar or nonpolar, so they attract or repel each other to form kinks and folds in the proteins. And having these polar and nonpolar features can make proteins function as like integral structures in cell membranes. So back to the cell membrane. If we have an integral protein that goes all the way through the cell membrane, like a transport channel or something, there'll be part of the protein that's nonpolar, so it can interact with fatty acids, and it will have part of it that's polar, so it can interact with like the phosphate head, and then whatever ions, it's transporting through the membrane. So proteins are what make up so many structures of your body, and they perform lots of functions. So structural proteins, we'll talk about like collagen, and functional proteins will be like enzymes, things like that. Okay, so the figure 2.26 shows representative amino acids. The figure depicts different types of functional groups. Each amino acid differs from the other amino acids in the number and arrangement of atoms in their functional groups. So we see that we've got nonpolar amino acids and polar amino acids, as we had mentioned earlier, which gives them all kinds of behaviors. So, how to make a protein? When amino acids are joined, a hydrogen ion is stripped from the amino end, and a hydroxide is stripped from the carboxyl end. And so you might be like, oh, that's dehydration synthesis. And that is correct. It sure is. And so the resulting bond is called a peptide bond. When amino acids are added together to form a chain, it is the primary structure of the protein. That also can be called like a polypeptide chain. Figure 2.27 shows the formation of peptide bonds by dehydration synthesis reaction. So we're losing the hydroxide group from, looks like the carboxyl portion, and the hydrogen comes from the amino portion. And then the resulting formation is a peptide bond. And it looks like we bonded 1, 2, 3, 4 amino acids together and formed 3 peptide bonds, and we got 3 waters as a result of that dehydration synthesis. So you're starting to see why the body is made up of all this water. Not only does it have electrolytes dissolved in it, and we have intracellular and extracellular fluid, but we also use water when building and breaking down bonds. Okay, so a chain of amino acids, that primary structure is called a polypeptide chain, and it can vary in length from 3 to 4,500 amino acids. And the chain is called the primary structure of the protein, and the sequence of amino acids in a chain is determined by the DNA. That's what genes code for, is the polypeptide sequence. Weak hydrogen bonds may form between neighboring amino acids. This may form an alpha helix or a beta pleated sheet, and this is called the secondary structure of the protein. This is illustrated in figure 2.28. A is the primary structure, the polypeptide strand. And it's the sequence of amino acids in that polypeptide chain. The secondary structure in B is the conformation of the chain created by hydrogen bonding between amino acids. This could either be an alpha helix in B here, or a beta pleated sheet in C. Where's C? There it is. The tertiary structure, which is D, is the three-dimensional structure of the protein. The formation of a protein by the bonding of two or more polypeptide chains would be the quaternary structure of the protein. Hemoglobin, the protein in red blood cells that carries oxygen, is used here as an example. Each heme group contains a central iron surrounded by a flat organic molecular structure. And the hemoglobin is very important, as we saw, in transporting oxygen. And if one amino acid is out of place, then the hemoglobin molecule becomes warped and the red blood cell becomes sickle-shaped. Thus, a attraction to amino acids further away produces bends and folds, creating a specific 3D shape. And that shape is critical in dictating function. So this goes back to that structure dictates function. So since weak bonds hold tertiary structures together, a protein is easily denatured or unfolded by changes in pH or temperature. Some tertiary structures are made more stable by disulfide bonds between cysteine amino acids. Figure 2.29 shows the bonds responsible for the tertiary structure of the protein. The tertiary structure of a protein is held in place by a variety of bonds. These include relatively weak bonds such as hydrogen bonds, ionic bonds, and van der Waals or hydrophobic forces, as well as the strong covalent disulfide bonds. It says that it was the cysteine amino acid that showed those together, or that bonded those together. Some functional proteins are composed of multiple polypeptide chains covalently bonded together, and this would be called the quaternary structure of the protein. So the examples are the hemoglobin, which is 4 polypeptides bonded together, and insulin is 2 polypeptides bonded together. Conjugated proteins. Sometimes proteins are combined with other molecules. I love these, like the glycoproteins. So that's going to be a protein and a carbohydrate. And there will be hormones and also some cell membranes. Back to the cell membrane here. We talked already about phospholipids, cholesterol, integral proteins, and now we're talking about glycoproteins. And so the glycoproteins in the cell membrane are important for cellular communication. They're how your immune system recognizes your cells as yours. And then we have lipoproteins, which are a protein plus a lipid. And so these are also going to be found in our cells membranes, and they are going to be carrier molecules in the blood, such as like those me cells and the LDL cholesterol and the HDL cholesterol. Sorry, tongue-tied there. Protein functions. We've got the structural proteins, and we've got those enzymes, as I had mentioned. So structural proteins are collagen fibers in connective tissues, keratin in the skin. So these are largely fibrous proteins in nature, and their importance cannot be understated. Collagen fiber binds all of our organs and tissues together. If you were to remove the living cells in an organ, you would still see the outline if the collagen was left behind. You could see every feature of that organ. So it basically is like the foundation of how our body is built, is that collagen fibers. And then keratin, of course, waterproofs your skin, as we had learned in chapter 1. It adds strength and resiliency to your skin, and it also aids in protection and forms your hair and nails. So a lot of these other proteins are what we call globular proteins, meaning that they're, globe-shaped, circular, 3D circular shape. And so enzymes assist in every chemical process in the body, such as the dehydration, synthesis, and hydrolysis. Antibodies are important in the immune system. They flag, bacteria for destruction. We've got receptors, which receive communication from other cells for regulation of cell activity. And this could be like for hormonal activity, neurotransmitter activity, etc. And then we've got carriers, like those integral membranes or integral proteins that go across cell membranes. And then we also have carriers in our blood, particularly to carry lipids. Because the reason we need carriers for lipids in the blood is since lipids are nonpolar and plasma is mostly water, the lipids can't just simply float around in blood to go from place to place. All right, this is figure 2.330, and it is a photomicrograph of collagen fibers within connective tissue. Those big pink ones, and it's like steel beams. The collagen is as strong as the steel of the similar diameter. And then the elastic fibers do that whole stretch and recoil. Recoiling is what is the elasticity part, not the stretch. It's the recoil. Okay, well, we've reached the last biological molecule, which I know you're saying hallelujah because you're tired of me talking to you. But let's just talk about nucleic acids, and then I'll let you go on with your day. So nucleotides are the building blocks for nucleic acids, and they are composed of a 5-carbon sugar. So that's that pentose sugar, 5-penta. They're also got a phosphate group, that PO4 negative group, and they have a nitrogenous base. So the nitrogenous bases are going to fall into 2 categories, a pyrimidine or a purine. A pyrimidine is a single carbon ring plus nitrogen. And I think of that as like a pyramid is like one structure. So a pyrimidine is a single carbon ring, and it has a nitrogen added to it. And so examples of that are cytosine, thymine, and uracil. A purine is 2 carbon rings plus a nitrogen group, and these are going to be guanine and adenine. The pentose sugars are either deoxyribose or ribose. So you may guess DNA has deoxyribose and RNA has ribose. So let's talk about deoxyribonucleic acid, or DNA, first. The sugar in this molecule is called deoxyribose and can bind to one of 4 nitrogenous bases: guanine, thymine, cytosine, and adenine. Deoxyribose bonds with a phosphate group via dehydration synthesis to form a long chain, which serves as the backbone of the molecule. So we call this a sugar-phosphate backbone of DNA. And so the nitrogenous bases are going to be attached to that sugar-phosphate backbone, and the nitrogenous bases are going to bind to each other. So each nitrogenous base can form a hydrogen bond with another to result in a double-stranded molecule. Cytosine can only bind with guanine. Thymine can only bind with adenine. So this is the law of complementarity base pairs. And I said that weird. The law of complementary base pairs. I was saying like complementarity of structure and function. Anyway, it's the same kind of thing, complementarity of base pairs. So it's very specific. C and G T, and A. The two chains of DNA twist, and this forms a double helix, and DNA is the basis of the genetic code. The sequence of bases code for amino acids to make a protein, more specifically a polypeptide chain. And then the hydrogen bonds and polarity of those amino acids are what fold and bend the protein into the secondary and tertiary structures. Okay, so this image, B2.31, is the structure of nucleic acids, and the components of a single nucleotide is the top, and the structure of the polynucleotide is found below. The polynucleotide was formed by dehydration synthesis reactions between nucleotides that join the nucleotides together by sugar phosphate bonds. And this is important because this is occurring all the time. When we learn about like RNA and all kinds of stuff, we're constantly building templates off of DNA, and during like DNA synthesis, when we have to duplicate the DNA to then divide our cells by mitosis to grow, that's going to require the building of these nucleic acids. So once again, water and dehydration synthesis is extremely important. Now we're looking at figure 2.32, which shows the structure of the 4 nitrogenous bases. So the cytosine and the thymine are the pyrimidines, because remember they're single structures, like a pyramid is single. And then the purines are the guanine and adenine. And then this shows that they're very specific in which base pairing they can do. The guanine can bind to cytosine, the thymine can bind to adenine, and that's the way it is. Figure 2.33 shows the double helix structure of DNA. The two strands are held together by hydrogen bonds between complementary bases in each strand. And these hydrogen bonds are easily broken and formed, as we mentioned earlier, and so they've got the dashed lines here. And so that, again, it's very important because we're constantly like zipping up DNA and unzipping DNA in order to make RNA molecule templates off of the templates. We also need to then sometimes, synthesize and duplicate our DNA in order to grow and reproduce. So let's talk a little bit more about RNA. RNA is an unsung hero. It's similar to DNA, except it has ribose sugar instead of deoxyribose. It is single-stranded instead of double-stranded, and it has uracil instead of thymine. The differences between the nucleotides and sugars in DNA and RNA is featured here in figure 2.34. DNA has deoxyribose and thymine. So deoxy means without the oxygen there. So deoxyribose and a thymine. Whereas ribose has a ribose sugar. It has that oxygen attached and a uracil instead of thymine. It's thought that RNA evolved prior to DNA, being a simpler structure, etc., and may have been the first like hereditary molecule in early protocells. And it still plays a vital role in the function of the DNA. There are 3 types of RNA that are used to take information for assembling a protein out of the nucleus and actually assemble it. So this is going to be where the DNA is going to unzip at certain points, and like a messenger RNA will be built from that DNA template. The messenger RNA can then leave the nucleus and go out into the cytoplasm, and there it is going to meet up with a ribosomal RNA. Ribosomes are going to be what we call the protein factories of the cell, because they are able to read the messenger RNA and attach the transfer RNAs to the messenger RNA and build the protein template off of it. It's a fantastic process, one of my entire favorite processes of all of biology, protein synthesis. But we don't have to really go into that, so we can get to the other types of RNA, which are amazing as well. So we've got ATP and GTP, so adenosine triphosphate, and then we've got GTP, which it doesn't spell that out there, but it also talks about CAMP, which is called cyclic AMP, and that one we'll learn about when we get into the hormones. And then we have NAD, which is nicotinamide adenine dinucleotide, and FAD, which is flavine adenine dinucleotide. And these are going to be very important coenzymes in the cellular respiration used to get our energy from our food. So the NAD and FAD are ultimately responsible for synthesizing ATP in our cells, and then cyclic AMP is important for hormonal regulation in many cases. And that concludes chapter 2. Thank you for listening. I hope that I did a decent job for you all.